Chapter 13 is meiosis. Now, we've already finished discussing Chapter 12, mitosis. Mitosis is ordinary cell division, necessary for growth, maintenance, and repair of organisms and their tissues. Meiosis, however, is a type of cell division for reproduction. So this serves as a bridge between the chapter on cell division and the future chapters on heredity. Now, to understand the material from Chapter 13, it is essential that you understand the material from Chapter 12. It is going to be very hard to understand meiosis unless you already understand mitosis. Okay? So if you don't yet understand mitosis, you need to go over that some more. Now, meiosis is mainly for reproduction. Now, in our notes on Roman numeral 1, I sort of introduced the concept of heredity, genetics, DNA, chromosomes again. Now, our chromosomes are paired in our body cells. That's because we have two parents. In human body cells, we have 46 chromosomes arranged as 23 pairs. Each set came from each parent. One set came from mom, one set came from dad. So that is referred to as diploid. 2N is the symbol for diploid in most organisms. Now N is haploid, half the diploid number. Okay? So for humans, 2N is 46 and N is 23. So our body cells are diploid, 46 chromosomes. Page 250 in your textbook shows you some photograph of chromosomes. This is called a karyotype. A karyotype is a procedure to view chromosomes. Now, in Roman numeral 2 of our notes, I compare and contrast sexual and asexual reproduction. With asexual reproduction, an organism gets all of its genes from a single parent. There is no genetic recombination because the offspring is basically a clone of the parent. With sexual reproduction, what they have to do is they have to divide the number of chromosomes to produce egg and sperm cells, and then as they fertilize, you have genetic recombination of the offspring. So in sexual reproduction, an organism gets its genes from two parents, and the organism is not genetically identical to the mother or the father or necessarily even the siblings. So that's the biological advantage, maintaining variability of a species. Roman numeral 3 of our notes, an introduction to sexual life cycles. For this you can look at figure 13.5. What happens is that a diploid parent will produce gametes, egg cells and sperm cells, from the organism through what is called meiosis. So gametes are haploid. Okay? So egg cells are haploid, sperm cells are haploid. When they fertilize, you now have a zygote, a fertilized egg, which is now diploid. And then that becomes a new diploid individual. I talk a little bit about chromosomes. Somatic cells are body cells. They have diploid chromosome sets, humans 46. Now, of our 23 pairs, 22 of those pairs are called autosomes, and one pair are sex chromosomes. The sex chromosomes are the X and the Y chromosomes. XX for female, XY for male. We'll talk about that in a later chapter again. Now, each pair of chromosomes is called a pair of homologous chromosomes, one from each parent. Okay? So make sure you cover these terms under Roman numeral 3. Now, Roman numeral 4 covers meiosis itself. Pages 254, 255 in your textbook shows you the pictures. Pay special attention to the colors of the chromosomes. Okay? The colors of the chromosomes are depicting one from each set. So with each homologous pair, you're going to see a red one and a blue one. Okay? The drawings on pages 254 and 255 show you an organism that has 6 chromosomes arranged as 3 pairs. Okay? So the haploid number is 3, the diploid number is 6 in this example. Now, with meiosis, there are 2 divisions, meiosis 1 and meiosis 2. You start off with a diploid parent cell, you end up with 4 haploid daughter cells. The daughter cells are not genetically identical to the original parent cell, nor are they genetically identical to each other. Okay, so that's the difference between meiosis and what you learned in the previous chapter. Now, each meiosis division has its own prophase, metaphase, anaphase, and telophase. But let me go over this very quickly. Now, with prophase 1 of the first meiosis division, nothing new to tell you there other than the ordinary prophase events. But one additional thing happens in prophase 1 of meiosis. The pairs, the homologous pairs of chromosomes come together, forming what are called tetrads, and they will actually exchange pieces with each other. That's called crossing over. You'll notice that a piece of a red and a blue switch on the pairs. That happens in prophase 1. With metaphase 1, the chromosomes line up. Not in single file as they did in mitosis, but they line up as homologous pairs. So this is different. So anaphase 1 will also be different. The homologous pairs separate, not the sister chromatids. So it's anaphase 1 that you're actually separating the pairs of chromosomes. So after telophase 1, we're now having 2 daughter cells that are haploid. So the first division separates the chromosome pairs. The second division is going to separate the sister chromatids. So really it's meiosis 2 that is much more similar to mitosis. So again, prophase 2, nothing new to tell you there. Metaphase 2, chromosomes line up just like they did in mitosis. Anaphase 2, centromere splits, sister chromatids separate just like in mitosis. And after telophase 2, we now have a grand total of 4 haploid daughter cells. Now, as I mentioned just a second ago, these daughter cells are not genetically identical to each other. That's what allows for genetic recombination. That's what allows for genetic variability of an organism, of a population, of a species. Now, population variability is vital so that natural selection can have something to select from, making adaptation possible. For this part, you need to be looking on page, well, let's see. Look on pages 258, 259. Now, again, the notes won't make much sense to you without looking at the pictures, okay? So figure 13.11 shows you what's called independent assortment of homologous chromosomes. So what it means is that during metaphase 1, each pair of chromosomes lines up independently at random with every other pair. Now in this particular example in figure 13.11, it shows you 2 pairs of chromosomes, giving you 4 possible outcomes of the gametes just with independent assortment of homologous chromosomes. Now there's a formula there. That formula is 2 to the n power. Okay? Where n is the haploid number. So in this case, the haploid number was 2, and it gave you 2 to the 2 power, which is 4 possible combinations of the gametes. That is what chromosomes they're getting from each pair based on independent assortment alone. Now for humans, that's 2 to the 23rd power. Sorry about that. Which is over 8 million possible combinations based on independent assortment alone. So that's one source of genetic variation, independent assortment during metaphase 1. Another source of genetic variation is random fertilization. Okay? So if each egg cell represents 1 in over 8 million possible combinations for humans, and each sperm cell represents 1 in over 8 million possible combinations, that's 2 to the n times 2 to the n for the zygote. That's over 70 trillion possible combinations. Okay? What that means in a nutshell is that if you and your mate have 70 trillion children, 2 of them might be genetically identical, but wait, there's more. Now, these numbers do not take into account crossing over. That adds to the genetic variation even further. By how much? Who knows? If only one crossover event occurred in the whole thing, that already doubles your number. Okay? So if that's random, we're talking hundreds of trillions to 1 in terms of the amount of genetic variation that is available. Very staggering indeed. And that finishes chapter 13.